Okay, so welcome back from the break. Let's take a look at the two concept check questions so we can move on to the third and the shortest section of this chapter. So identify the two major functions of pol 3 in DNA replication. So its first function is to attach, attach new nucleotides to the 3' end of the growing complementary strand. So that's job 1, and job 2 is and proofread. So that's the two jobs that it does. It proofreads, it adds the new nucleotides in, and as it's doing that, it's proofreading to make sure that the correct one is being added on each occasion. So if pol 1 in a cell was non-functional, what effect would that have on the synthesis of the leading strand? We know what its function is on the lagging strand. It's the one that comes in and removes the primers from the Okazaki fragments so that the fragments can be connected together by ligase after pol 1 inserts the appropriate DNA nucleotides. But does it have a role in the leading strand? Well, the answer is yes. And to understand why, we need to go back to an earlier diagram, this diagram. So we can see here that pol 1 is replacing the nucleotides in a primer. But remember, up here on the leading strand, there's going to be a primer in here. This, of course, is an Okazaki fragment. Because if you look at the overview diagram, if this is the origin of replication with its primer, then this is an Okazaki fragment. So when this is copied by DNA pol 3, it's going to eventually run up against this primer. And it's going to have to stop because it doesn't know what to do with that primer. But pol 1 does. So this is the role of pol 1 on the leading strand. It's to take out the primer at the origin of replication and replace the DNA nucleotides, or the RNA nucleotides with DNA nucleotides, so that ligase can link up these Okazaki fragments to this leading strand fragment. And if pol 1 was not active, those primers would stay in place. So while we typically think of pol 1 being active in the lagging strand, we have to remember that there are still primers at each of the origins of replications that represent the beginning of the leading strand replication that are going to have to be removed at some point along the way as the Okazaki fragments of the adjacent lagging strand come up to that origin of replication and therefore need to be linked up to it. And that's a job for pol 1 and ligase, just as we saw on the lagging strand copying process. So we can't forget that DNA pol 1 works on both of the strands, not just the lagging strand where we might initially think that its role is, because there are primers on the leading strands that also need to be removed. They're located at the origins of replication. So the last little section talks about the packaging of DNA. So in the case of bacterial DNA, it is a circular DNA molecule that is supercoiled so that it can fit inside the bacterial cell in a region known as the nucleoid. When we disrupt a bacterial cell, like an E. coli cell, you get a really clear indication of just how important that supercoiling packaging actually is, because here is the single circular DNA molecule of this E. coli, and you can see that it is huge in terms of the space that it occupies compared to the cell that it was packaged into. So we've disrupted the supercoiling, we've actually disrupted the internal structure of this cell so that you can get an idea as to just how big the DNA molecule is compared to the bacterial cell. So you can see the advantage of supercoiling. You can see that there are some tiny little circular DNA molecules scattered around. These are the plasmids. These are the auxiliary circular DNA molecules. These are the location of things like antibiotic resistance genes are typically associated with these. Some genes that are involved in transferring DNA directly from bacterial cell to bacterial cell by a process that would be as close as you can get to something like bacterial sex. It's driven by specialized plasmids called F plasmids that allow for the exchange of DNA directly from one bacterial cell to another via an actual physical connection between the two bacterial cells. And they don't have to be bacterial cells of the same species. That's the scary thing here. When bacteria exchange DNA, they don't necessarily have to do it with members of the same species. They can do it with any bacterial cell. That's what makes antibiotic resistance so scary, because of where it's located. In the case of eukaryotic cells, of course, we know that the DNA there is much bigger. You know, human DNA, if it was chromosomes, if they were linked end to end, would stretch nearly 10 feet. But one way in which we can package them inside the nucleus is simply by breaking them up into smaller units. So we have the 46 linear molecules, we call them chromosomes. Then we know that those molecules are then complexed with proteins called histones. And those histones form complexes that the DNA wraps around. And that means that we now shorten the length of the linear molecules. But then those linear molecules are themselves then complexed into either 10 nanometer DNA, which is the form in which it is found with the histones, and this is referred to as euchromatin. But it can also be then initially coiled into what is known as a 30 nanometer fiber, which is also chromatin, but this is known as heterochromatin. So in the form of in its euchromatin form, it's at least accessible for transcription. So the genes here would be available for activation, for transcription. They're certainly available for activation if it's in the 2 nanometer form, but it's also in the 10 nanometer form. It can be active for transcription. In heterochromatin, it's inaccessible. Transcription. And you might find this in the telomere regions. You might find this also in the centromere regions. The DNA is in the form of the 30 nanometer fiber where it's been inactivated and not available for transcription. Then, of course, we know that it can be further folded into this loop and coil structure, which gives you the highly condensed chromosome that then becomes the 2 sister chromatids in the duplicated chromosome that you can see during cell division when the chromosomes are condensed and visible. So I've told you a little bit about all of this. We do know that the chromosomes tend to occupy specific regions within the nucleus. So you can take the chromosomes when they are in their condensed form and you can quote unquote paint them so that you can identify the homologous pairs, because each member of the pair is the same, as an identical color. And if you allow those chromosomes then to occupy a nucleus, you can see that each of the chromosomes occupies a specific region. They tend not to overlap, but what is even more noticeable is that the two members of a homologous pair do not necessarily occupy adjacent regions. They could be anywhere scattered throughout the nucleus. We do know that within the nucleus there is evidence of a skeleton similar to the cytoskeleton inside the cytoplasm, and there's even some evidence for something equivalent to the cytoplasm, which is referred to as nucleoplasm. And this all would provide a scaffold inside the nucleus for the anchoring of the chromosomes in their specific regions within the nucleus, and probably also serves in some fashion to control whether they're in the 2 nanometer, 10 nanometer, or 30 nanometer form of the DNA molecule, which in turn helps to regulate the expression of the genes in the different locations within the chromosome. So I've told you a little bit about euchromatin and heterochromatin, so I won't repeat that. I gave that to you in a couple of slides prior to that. So that brings us to the end of the body of the presentation. I told you this last section would be quite short, but here are a couple more concept check questions just to finish off this section. So what two properties, one structural and one functional, differentiate euchromatin and heterochromatin? And if you stopped the human cell in metaphase 1 and painted the two homologs of each homologous pair the same color, what would you expect to see? And how would this differ if you did the same thing in metaphase in mitosis? So let's look at the first question. What two properties, one structural, one functional, differentiate euchromatin and heterochromatin? Well, remember, euchromatin is the 10 nanometer form. And in the 10 nanometer form, complexed with the histones, the DNA is available or accessible for transcription. So there's the potential for the genes there to be activated, to be transcribed. In the heterochromatin form, remember, that's the 30 nanometer form. And in that form, it is not available for transcription. So the genes are inaccessible, and that would be the centromere regions, the telomere regions, and any genes that are inactive in the cells. They would tend to be in regions that are in heterochromatin form. So if you stopped a cell in metaphase and painted the homologs the same color, what would you expect to see? Well, in metaphase 1, remember, the homologous pairs of chromosomes line up on the metaphase plate. So what you would expect to see is pairs of chromosomes of identical color lining up on the metaphase plate, and you'd be able to identify the chromosomes as homologous pairs because they would be the same color and they would straddle the metaphase plate, being the same color and being members of the same homologous pair. How would this differ in metaphase in mitosis? Well, it would be the individual chromosomes that will line up on the metaphase plate, and they may not necessarily be adjacent to each other as homologous chromosomes. They may be located anywhere along the metaphase plate. They may not necessarily line up in the same location as their homolog because there's no necessity for them to do so. They simply need to line up as individual chromosomes on the metaphase plate because remember, all that's going to happen here is that the sister chromatids are going to separate for each individual chromosome. Whereas in metaphase 1, the two homologs need to line up together so that one homolog goes in one direction, the other homolog goes in the other direction, because in anaphase 1, it's the homologous pairs that separate. So it's going to be the homologous pairs painted the same color that are going to line up on the metaphase plate. So that's it for this presentation. It's been somewhat long, and I realize that the first two sections are somewhat long, but take your time going through them, and hopefully you get to this point having being able to stop and start and think through what you're seeing there and being able to make some good notes and feel confident that you understand the process of DNA replication and also the structure of the DNA molecule. But if you have any questions, concerns, or comments about anything that I've included here, by all means, come and see me during office hours and let's clarify any issues that you have. Otherwise, I'll see you back here for the final presentation for the semester when we talk about transcription and translation. See you then. Bye.